Hello everyone, welcome to VLearn and welcome back to the videos of Introduction to the Genetics part. In today's video, we are going to learn about the post-Mendelian genetics. In the post-Mendelian genetics, that is the period after the Mendelian classic genetics. If we talk about the classic Mendelian genetics, we have studied the principle of inheritances, which were basically purely given by Mendel. We have studied principle of segregations, principle of paired factor, principle of dominance, and principle of independence, principle of independent assortment. In today's class, we will study about the post-Mendelian era. If we study post-Mendelian era, we will study basically two parts. We will basically study in two parts. So if we talk about the post-Mendelian genetics, post-Mendelian era of genetics, it is basically divided into two parts. The first part is intragenic inheritance. Intragenic inheritance. The other part is intergenic inheritance. What is intragenic and intergenic inheritance? Intragenic inheritance is also known as interallelic inheritance. Interallelic inheritance. Intra means within. Intra means within. Within the gene. That means if we talk about a gene have two alleles. A gene have two alleles. That is, this inheritance, this type of inheritance basically occur within the same gene. So this type of inheritance basically occurs within the same gene. Same gene, or you can say between two alleles of the same gene, or two alleles of same gene. So basically, if we talk about one gene have basically two alleles. So it is basically the interaction between these two different alleles. So basically, it interacts to produce these two alleles. These basically two alleles interacts to produce particular phenotypic expression. So this interact to produce phenotypic expression. The other part is intergenic inheritance. What is intergenic inheritance? If we talk about intergenic inheritance is basically between two genes, or maybe greater than two genes. At least two different genes would be involved in this kind of inheritance. Of course, if we are talking about these two genes or more than two genes, the inheritance, this type of inheritance typically occurs between more than two or two different genes. These two or more different genes interacts to produce different phenotypic expression. So basically, if we talk about intragenic inheritance and intergenic inheritance means between one gene, between two alleles of the same gene, But if we talk about the intergenic inheritance is between two genes, not between the alleles anymore, or it can be between more than two genes, like polygenic inheritance. So these two or more than two genes interact with each other to produce the different phenotypic expression. So basically, we will study the intragenic and intergenic inheritance now. The first kind of intragenic inheritance is incomplete dominance. So let us now study incomplete dominance. Incomplete dominance was basically first discovered by Carl Correns. So Carl Correns was the one who basically first observed this phenomena of incomplete dominance. Incomplete dominance was basically observed in the plant Mirabilis jalafa. Mirabilis jalafa, which is also known as 4 o'clock plant. The other plant in this is Antirrhinum majus. So basically he was studying with Mirabilis jalafa and Antirrhinum majus. What he did was, he took homozygous red flower. He was studying the same experiment studied by the Mendel. So he was basically studying homozygous red flower plant. Homozygous red flower. He obtained the homozygous red flower and crossed it with homozygous white flowers. Homozygous white flowers. Basically, let us consider if the red is dominant, white is recessive. The result should be all the flowers in the F1 generation should be red if we talk about the Mendelian concept of inheritance. Or if the white flower is the dominant one, then all the flower in the F1 generation should be white color. So basically, when he did his experiment, that means Carl Correns was doing this experiment with the plant what? Mirabilis jalafa. He was expecting to get either the red flower or the white flower in the F1 generation. The expectation was either red flower or white flower he will be getting in F1 generation. So this is expectation. So with this expectation he did crossed. After obtaining the results of the cross, in the F1 generation he was totally surprised. Neither of the flower was red or white. He was totally surprised. He thought maybe the experiment went wrong. He did the same experiment again, but he got the same results. And the result were all flower in this Mirabilis jalafa plant were pink. None of the flower was basically red or white. That is, neither of these two alleles are dominant. That is, this can only happen if neither of allele is dominant. Therefore, this F1 generation is neither white nor red. Instead, this F1 generation is intermediate of the two. Therefore, all the plants in F1 generations were pink. He did the same experiment, that is, all the red flowers crossed with the white flowers obtained the pink flowers in F1 generation. This was the first step. Second step was cross-pollination, obtained the F1 generation the pink flowers. Then he self-pollinated this. Self-cross. Upon selfing the pink flowers in the F2 generation, let us say the red color is basically responsible for the R1 gene. So R1 gene is basically responsible for color red, whereas the W1 gene is basically responsible for white. So the genotype of this plant would be RW. So RW was basically crossed with RW. So if we talk about the gametes, the one gametes would be R1, W1. The other gametes of the male plant would be R21, W1. So if we cross R1 with W1, the result would be R1, 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 W1, R1, W1, and W1, W1. The plants with this genotype would be, of course, red. The plant with R1, W1 will be, of course, pink. So this was pink, this was pink. The plant with W1, W1 will be, of course, white. The normal ratio, the normal phenotypic ratio, the normal phenotypic ratio of F2 generation according to Mendel was 3:1. But the normal ratio of 3:1 is changed from 3:1 to 1:2:1 because one of them is red, two of them is pink, and one of them is white. So the normal ratio of this plant is changed from red is to pink is to white. The normal 3:1, that is phenotypic expression ratio, is changed from 3:1 to the 1:2:1. And same is with the genotypic ratio. That means you can say that the phenotypic ratio and genotypic ratio for incomplete dominance is same. The phenotypic ratio is 1:2:1. The genotypic ratio is also 1:2:1. So the phenotypic ratio, which is normally 3:1, is changed to 1:2:1, whereas the genotypic ratio is changed to genotypic ratio doesn't change, genotypic ratio is same. Or you can say the phenotypic ratio and genotypic ratio for the incomplete dominance is 1:2:1. So this is the incomplete dominance, and what we got from the incomplete dominance is that the phenotypic and genotypic ratio in F2 generation is same, and it is 1:2:1. The phenotypic ratio of 3:1 is changed to 1:2:1. So this is the case with incomplete dominance. In the next class, we will solve some numerical problems related to the incomplete dominance. So next class is all about the numerical problems related to the incomplete dominance and the principle of the inheritance. Thanks for watching the video. I hope you love the video. If you have any doubts after watching the video, please mention that in the comment section. I will reach out to every comment and hope that I will be providing the solution, the detailed solutions of all the problems after reading the comments. Please also like and subscribe our channel for more informational videos on genetics and other biology subjects. See you soon in the next video. Thank you for watching.